Welcome to this explainer. Today, we're looking at something absolutely mind-blowing. Scientists have actually figured out how to literally bottle nature's living light to solve some incredibly complex human problems. It's a total masterclass in biomimicry, where ancient biology meets cutting-edge technology. Let's dive right in. Here's our roadmap for today's explainer. We'll start with nature's living lights, dive into the chemistry of glow, see how we're harnessing firefly power, move on to catching contamination with light, and finally, wrap things up with bacterial bioluminescence. All right, Section 1, Nature's Living Lights. So how exactly does nature create light in the pitch dark? When we humans need light, we flip a switch for electricity, or we burn something to generate heat, like fire. But nature? Nature figured out how to do this completely cold, super efficiently, and entirely from within. Well, the answer is right in front of us. Take a look at that brilliant blue glow in these test tubes. That is living a liver brio fishery bacteria. And they aren't, just soaking up sunlight and reflecting it back. They're actually using a highly specialized protein-driven process to manufacture that light completely from scratch. Which brings us to Section 2, the chemistry of glow. Okay, to really get this, we need to meet the two main characters here, luciferase and luciferin. I want you to think of this whole setup like a biochemical engine. Luciferase, that's the heavy-duty engine itself. It's the protein catalyst driving the whole reaction. And luciferin is the intricate machinery inside, the actual small organic compound that physically emits the light once it gets oxidized. This side-by-side view really nails it. On the left, you've got the macro, the beautiful firefly we all recognize. And on the right, the microscopic chemistry making the magic happen. Now, every luminescent species has its own unique toolkit. Fireflies rely on a specific structure called D-luciferin. When that D-luciferin oxidizes, it actually transforms into oxyluciferin, and boom, that exact chemical shift gives off that signature, beautiful yellow glow. But hey, an engine can't run without fuel, right? For this specific reaction, the fuel is ATP, along with oxygen. No ATP means absolutely no reaction, which means none of that highly specific 562 nanometer yellow light. It's this perfectly balanced equation. Energy goes in, and precise measurable light comes out. Let's move to Section 3, Harnessing Firefly Power. This quote hits the nail on the head regarding why this is an absolute game changer. Since literally every living cell on Earth relies on ATP for energy, it's basically a universal footprint for life. By detecting even attomole levels of ATP, and just to put that into perspective, an attomole is 10 to the negative 18th power. It is an incomprehensibly tiny amount. By finding even that microscopic trace, you can detect invisible, potentially dangerous living microbes. And that exact realization led scientists to create advanced systems like the Enlighten biosensor. It literally uses luciferase derived from wild fireflies, which, as you can probably guess, is pretty expensive. But it uses it as this invisible biological detective to hunt down hidden bacteria in our hospitals and our food supply. All right, Section 4, Catching Contamination with Light. So how does it actually work? Well, the real genius of this workflow is Step 2, which is lysis. See, you can't just rub luciferase on a counter and expect it to glow. First, you have to use special enzymes to forcefully break open or lyse the hidden bacteria. Smashing that outer membrane open is what releases the crucial ATP fuel we need to kickstart that light reaction. If you follow along with this schematic, you see the whole thing from start to finish. First, the swab is soaked and rubbed all over the suspect's surface. Then, you pop it into a tube, add the extractant to bust those cells wide open, and finally, drop in the glowing reagent. You can literally read the luminescence right then and there. It's incredibly fast. Here's what it looks like in practice. The technician mixes the free suspension of luciferase right there in the tube, and then pops it into that sleek black device you see on the left. That's a luminometer. It's a highly sensitive piece of equipment specifically designed to instantly measure the exact intensity of that flash of light. So why do we care about the exact intensity of the light? Well, as the standard curve clearly shows, the chemistry scales perfectly. It's a totally direct proportional relationship. More ATP simply equals more light, period. This gives quality control experts and scientists a rapid, highly accurate numerical count of exactly how much bacterial contamination is sitting on that surface. And finally, Section 5, Bacterial Bioluminescence. Take a look at the glow in this flask. When luminous bacteria like Allia fishery are grown in a nutrient-rich broth, they actually don't just start glowing right away. They wait. They wait until they reach a critical population mass, and then, boom. Once there's enough of them, they collectively trigger this continuous, mesmerizing glow of roughly 500 nanometer light. Now look at the contrast between these two biological engines side by side. Unlike the firefly, this bacterial system doesn't use luciferins at all. In fact, it completely ignores ATP. Instead, it uses a totally different compound called FMNH2 to generate its blue-green glow. It is just an absolutely incredible example of nature evolving two completely separate paths to achieve the exact same magic trick. Understanding both of these systems leaves us with a truly inspiring thought about biomimicry. I mean, think about it. We've literally taken an insect mating call and turned it into a crucial tool for hospital and food safety to protect human lives. It really makes you wonder, as we keep decoding these intricate biological engines, what other hidden, invisible truths might nature's flashlight illuminate next? Thanks for joining me for this explainer. Keep asking questions, and I'll catch you next time.